As you can see from this image, there are many different entities that can be studied by microbiologists. Some of them are cellular, such as the fungi and protists, bacteria and archaea, and some of these entities are considered acellular, such as viruses, viroids, and prions. And you can see that the organisms and biological entities that microbiologists study is kind of a wide variety, which means that defining microorganism or microbe can be a challenging thing to do. You have to encompass all of those different things that microbiologists study. And so the definition of microorganism that were used for the purposes of my course is organisms and acellular biological entities that are too small to be seen with the unaided eye. The most important part of this definition really is this unaided eye, right? This too small to be seen. And so anything that you really need a microscope to visualize is considered a microorganism. Whether or not that is considered kind of a live microorganism is still up for debate, right? So things that fall into that cellular category, such as bacteria and archaea, contain at least one cell, and cells are the structural functional unit of life. They can replicate by themselves. They don't need any additional help. And so things that fall in the cellular category are usually considered kind of live or organisms, whereas these acellular biological entities, such as viruses, can't replicate without the help of a host, and therefore are often considered kind of not alive. But both kind of live and non-live things can be considered microorganisms because they're unable to be seen without a microscope. And when we're talking about the cellular microorganisms, which is really the focus of this course, there are some kind of hallmarks of all cellular microorganisms. They are typically unicellular or made up of one cell. If they are multicellular, they do not have any differentiated tissues, and they're small. And small is also kind of a vague word, but in this case, small means anything less than 1 millimeter in diameter. And this size range for microorganisms can be relatively large. So there can be some that go up into 1 millimeter, and some that are more on the nanometer scale of size when you're thinking about viruses. So the size of microorganisms is pretty widely varied. And that's why sort of defining microorganisms is hard, and classifying them and grouping them into organized groups is hard as well. And so microorganisms were originally classified by the type of cellular structure that they had. And you can imagine that that kind of excludes all of those acellular biological entities, and then basically just groups all the cellular microorganisms into organisms that have a nucleus, or eukaryotes, versus organisms that don't. And this was sort of based on the fact that that was the best technology available at the time. Microscopes were okay, they could see cells. They could definitely see a very large structure in the cell, such as a nucleus. And so it was easy for scientists to classify cellular microorganisms into ones that didn't have nuclei versus ones that did. But, and so this led to this classification of all prokaryotes, or all of the non-nucleated cell organisms, into a kingdom called Monera. That was in the 5-kingdom system. So the other kingdoms were protists, fungi, animals, and plants. And Monera was this kingdom that basically all prokaryotes fell into. But we know that all prokaryotes are not created equal. And with the advent of new technologies, in particular DNA sequencing, taxonomists could move towards a more accurate way of grouping these organisms based on their characteristics. And they developed what's known as a 3-domain classification system, into which microorganisms fall. And so in terms of kind of the old groupings, you can see here the prokaryotes in blue and brown, and the eukaryotes in orange. And so the 3 domains of life are now considered bacteria, archaea, and eukarya. And you can see that the prokaryotes that used to be that kingdom Monera have now split into two groups. And that other biggest group is all of the eukaryotes. And what's interesting is you can find microorganisms in all three of the domains. So domain bacteria is what we consider this sort of true bacteria or eubacteria. Bacteria that you've typically heard of that can make you sick, such as Staph aureus. These organisms are all unicellular, or one cell, and they have cell walls with a particular structure and contain this molecule called peptidoglycan, which we'll talk a bit more about when we get to cell walls. Domain archaea, or the old bacteria, are also unicellular, like the members of domain bacteria, but they have a different cell wall structure. Their cell walls don't have this peptidoglycan, like the organisms in domain bacteria, and they have a unique lipid structure. And that is likely driven by kind of where we find archaea. They live in extreme environments. They're known as extremophiles, which can mean very high temperatures, very high salt, very low pH. And in order to deal with these extreme conditions, the archaea seem to have created some adaptations in their cell walls and membranes to really deal with their environment. And then finally, there's domain eukarya, which are just all organisms where their cells have nuclei. And both protists and fungi can be considered microorganisms, and they exist in domain eukarya.